Hey Team Bio. So today the screencast is on organic compounds, part 2. Last screencast, I talked about carbohydrates and fats and lipids, and today's screencast is going to be on proteins and nucleic acids. So, in the last screencast, we saw that all organic compounds are made up of carbon. Ooh, that's the wrong thing. Carbon, they also had nitrogen, hydrogen, and oxygen. In addition to these 4 elements, the molecules that we're going to look at today either have phosphorus, or sulfur. Okay, so the first type of organic compound, or organic molecule we are going to talk about is nucleic acids. And I'm sure you all have heard of nucleic acids before. 2 examples of nucleic acids are DNA or RNA. Deoxyribonucleic acid, or just straight up ribonucleic acid. Well, the function of nucleic acids are to encode genetic information. So that's what they do. And we're going to spend a long time this winter actually talking about how they encode that genetic information, but I'm not really going to address that in today's screencast. What I'm going to mostly focus on today is the structure of nucleic acids. So, I hope you all remember from the last screencast, I talked about the structure of monosaccharides. Well, the analogous structure of nucleic acids so a carbohydrate, the basic building block, is a monosaccharide. A nucleic acid, the basic building block, is called a nucleotide. So actually I'm going to put this under, let's make a new heading, call it structure. So a nucleotide is going to be the basic building block of our nucleic acids. And the structure of a nucleotide looks like this. It has a phosphate group, which I'm going to put in green, P, right here. And zoom out just a little bit. That phosphate is connected to a sugar, which is a 5-ring sugar, and that sugar is connected to what's called a nitrogenous base, and I'm going to color that in red. So let's label all these pieces here. This is the phosphate. Here is the sugar, and this is the nitrogen base. And this entire thing is a single nucleotide right here. This whole deal. Okay, so, however, in order to make a nucleic acid, what we actually have to do is make a long chain of these things. So I'm going to take this, shrink it down, and then, oops, put that in there. So, this would now be an example of a nucleic acid, a long chain of nucleotides. Now, there's something important, something special about these nitrogenous bases that I need to tell you. There are actually 4 of them. There's A and T, and then C, there we go, C and G. And they're all slightly different, but here's the interesting part about them. A is shaped in such a way that it only binds to T's. And C's are shaped in such a way that they only bind to G's. So what I've actually drawn for you right here would be a single strand of a nucleic acid, but in reality, DNA is always double-stranded, so I need to add in another strand to the other side, and I'm going to see if I can do that for you here. Okay, voila, did it. So, here's the deal. I erased all the notation. I hope you can still identify the phosphate and the sugar and the nitrogenous bases. But here, you notice I've drawn these all as being red. And this would never happen. Given my key over here, those would all be A's. So instead, what I should do to make this a little bit more accurate Okay, so now this is an accurate representation of what's going on here. Our A's are bonded to our T's, our C's are bonded to our G's, and additionally you should know that the bond between these molecules is a hydrogen bond. So I'm going to draw that in here as a dotted line. Hydrogen bonds hold these bases together. Okay, one last thing that I want you to notice is that these strands are running parallel to one another, but they're also running in the opposite direction. So all the oxygens and the phosphates are pointing up over here, and they're all pointing down over here. We call this anti-parallel. So, this is an example of a molecule of DNA, and it's running anti-parallel. Now, if I were to be drawing RNA, all I'd have to do is select half this molecule and erase it. And now, obviously this is not anti-parallel anymore because there's only one strand. And this would be an example of RNA. RNA is almost always single-stranded. DNA is almost always double-stranded. Okay. I think that's all you need to know about nucleic acids. So let's move on to proteins. I hate it when the ink is too skinny. That's better. Okay, so proteins. Proteins are made up of monomers, just like carbohydrates were made up of monosaccharides. Proteins are made up of amino acids. Now there are 20 different types of amino acids found in the human body. I'm going to paste them in here for you. Okay, so amino acids all have the same basic backbone. They're all made up of a carboxyl group right here, an amino group right here, but then they all have an R group. And here are the examples of all the different R groups of all the different amino acids. They're all different. So the differences between amino acids is the differences in their R group. And the different R groups give the amino acids different properties, like some are very polar and some are very nonpolar, which makes them either hydrophilic or hydrophobic. And these different properties are very important in determining their function, which I should talk about right now. So the function of proteins are multiple. There are many functions of proteins. Enzymatic functions. And don't worry if you don't know what an enzyme is or does. We're going to discuss this much more in depth later on in the year. They also have structural functions, so they provide support. They can also store things. I guess I should write storage. And lastly, they have hormonal functions. And they're responsible for cell-to-cell signaling. And you should know all of these functions are determined by the structure of the protein itself. Every different type of protein has a unique structure. Okay, so how do we make a protein? Well, there are a couple different steps. First of all, a single amino acid has to be linked to another amino acid, and so on and so forth, until we get a long chain. Okay, so here we have a long chain of amino acids, but you should know additionally that each bond between amino acids is called a peptide bond. It's a covalent bond, but we give it this special name, peptide bond. Okay, so many amino acids make what's called a polypeptide. So that's what we have right here, a polypeptide. But we still don't have a protein. This polypeptide needs to be folded in a very specific manner. And in order for you to understand this, I'm going to give you an analogy that will hopefully be helpful to you. Okay, so imagine that it is very cold outside. Okay, it's cold outside, and you're cold, so you decide you want to put on a sweater. But you don't have a sweater. However, you do have a sheep. Luckily for you, you have this sheep that has a lot of wool on it. So you shear the sheep, and you get this enormous pile of wool. But this wool alone is still not enough to keep you warm. You need something different than this. So you take the wool and you make it into a ball of yarn. So now you have yarn, and that's great, but it's still not enough to actually keep you warm. You need something with a very specific shape. So you knit a sweater, and voila. Now you put the sweater on, and you are warm. So the enormous ball of wool, that's analogous to the amino acids. The ball of yarn is analogous to our polypeptide. And it's only when we get to the sweater step that we have our protein. Polypeptides folded in a very, very specific shape. We call that shape a conformation in order to perform its function. Okay, I think that's all you need to know for now about proteins. So, boom, boom, this is it. Hopefully this was a little shorter. Indeed, it is a little shorter. It's only like 14 minutes.